In Chapter 9, we will look at what makes up DNA, how it compares to RNA, as well as how DNA is copied in the cell during cell division, transcribed, and then translated into a language proteins can read and use. Many scientists contributed to our knowledge of DNA, including Rosalind Franklin, who used X-ray technologies of the time to study DNA. It was her work that first showed images of the helical structure and the ladder-looking appearance of DNA. Chargaff determined the ladder rungs were composed of paired nucleotides, and later, Watson, along with Crick, took all of the work of several scientists and compiled it to propose a model for DNA. Watson and Crick also showed how DNA carried information and served as its own template for duplication. There are two types of nucleic acids, DNA and RNA. Both of these are made of strings or polymers of nucleotides. A nucleotide has three parts, a 5-carbon sugar, a phosphate group, and a nitrogenous base. On the 5-carbon sugar, each carbon is selective regarding what can attach at each spot. These 5 locations are referred to as primes. For example, the phosphate group is attached to the 5' carbon, while the nitrogenous base is attached to the 1' carbon. These locations are important to understanding the arrangement of DNA and RNA molecules. What makes DNA different from RNA? It's in how the two molecules are structured. They differ by only one oxygen molecule. The name of DNA is deoxyribonucleic acid, and it is composed of a 5-carbon sugar known as deoxyribose, while RNA's name is ribonucleic acid, and it is composed of a 5-carbon sugar called ribose. Notice in the image, on the left is ribose and the right is deoxyribose. They are the same except for in the red circle. RNA has an OH or hydroxyl group, while DNA has only a hydrogen atom. If you'll think of it as it has been deoxied. The location of this difference is at the 3' carbon. A phosphate group is a phosphorus atom bonded to 4 oxygen atoms. These groups attach to the 5' carbon of a sugar molecule in one nucleotide and the 3 carbon of the next nucleotide. Together, the phosphate group and the sugar form the backbone of a DNA strand. As discovered by Erwin Chargaff, there are 4 nitrogenous bases found in DNA. They form pairs due to their complementary structure and attach to the 1' carbon of the sugar molecule. Adenine and guanine are both a 2-ring structure known as purines, while thymine and cytosine are both a single-ring structure and known as the pyrimidines. In a complementary base pair, one purine bonds with one pyrimidine. Adenine and thymine always pair with each other, while guanine and cytosine always pair with each other. These base pairs are held together with hydrogen bonds. This image is a double-stranded DNA molecule. Notice inside the blue arrow starting at the top, there is a sugar molecule as indicated by the green pentagon, then a phosphate group as indicated in the orange circle with a P, then a sugar, then a phosphate, and so on. If you follow the blue arrow down, you will notice it alternates all the way down. These two parts are what are known as the backbone of DNA. They are the same all the way down and all the way up on both sides. Each set of phosphate group and sugar molecule is attached to one of the 4 nitrogenous bases as shown in the middle with the letters C, G, A, and T. These bases hold the 2 strands together using hydrogen bonds, which are indicated with the dots and the letter H. Notice how the phosphate group is bonded at one end to a 3 carbon of a sugar as indicated by the green arrow and the 5 carbon of the next sugar as indicated by the purple arrow. Each strand of DNA has a 5' end and a 3' end. This designation is directly related to which carbon of the sugar molecule the phosphate group is connected. The hot pink arrows at the top highlight the 5' end of the strand, as well as the 5' carbon where the phosphate group is attached. At the bottom, the 2 purple arrows indicate the 3 carbon where the phosphate group is attached and the resulting 3' designation of the end of the strand. When the 2 DNA strands are held together by hydrogen bonds, the little dots in the very center of this image, which hold together the nitrogenous bases, the G, C, A, and T in the middle, the DNA strands will run in opposite directions known as anti-parallel. Notice in the image on the left from top to bottom is 5' to 3', while on the right is the opposite 3' to 5' from top to bottom. As mentioned previously, nitrogenous bases are complementary to each other. A only pairs with T and G only pairs with C. Therefore, if one strand is 3' GT, GA, CAG, what would its complementary code be? If one strand of DNA is starting at the 3' GTGACAG, its complementary 5' to 3' sequence would be CACTGTC. These sequences carry coded information that tell the cell, and therefore the body, what and how to function. Watch the linked video for an animated version of DNA structure. As a reminder from chapters 6 and 7 on cell division, DNA is found in the nucleus of eukaryotic cells and in a region known as the nucleoid in the cytoplasm of prokaryotic cells. DNA is highly organized into chromosomes, which are inherited from parents. Chromosomes tightly wind genetic information so that it can all fit inside the cell. As DNA is replicated, these areas are unwound, so a copy of each strand can be made. On a strand of DNA are sections known as genes, as indicated in the red box, which have the unique sequences of base pairs that tell the cell what to do. When DNA replicates during the S phase of interphase, a copy is made of each strand. The strand serves as a template for replication due to the complementary nature of base pairs. Since both original strands are conserved in the replication process, this is known as semi-conservative replication. For DNA replication to occur, the 2 strands must be separated. An enzyme is used to unzip the double-stranded DNA. This enzyme is DNA helicase and works by breaking the hydrogen bonds between nitrogenous bases. As helicase separates the 2 strands, a replication fork is formed, exposing the base pairs on each strand. After helicase splits the 2 strands, a primer is needed for DNA to attach. Notice in the first strand in the image, only the original template half exists. As DNA can only add bases in the 5' to 3' direction, the primer provides an end for DNA bases to attach. Therefore, to initiate DNA synthesis, RNA adds a short primer of bases that can be used to start the DNA replication process. Once the short sequence of RNA bases has been added, another enzyme called DNA polymerase can then begin adding DNA bases to complement the original strand. As DNA replication begins in the replication fork, it will duplicate in both directions, which is known as bidirectional replication. Since DNA can only replicate in the 5' to the 3' direction, there are 2 types of duplicated strands formed. One can happen in a continuous fashion, which is called the leading strand. In this image, it is the top red strand, while the bottom strand has to be replicated in sections. This bottom strand, in this image is the bottom red with green strand, is called the lagging strand. An RNA primer will provide the end needed for DNA polymerase to begin attaching bases until it reaches the end of the section. These small replicated sections on the lagging strand are known as Okazaki fragments. Once replication has completed, all of the sections will be sealed off by another enzyme called DNA ligase. Watch the linked video for an animation of DNA replication. Note, however, the video includes several enzymes in more depth than we discussed. Telomeres, glowing in the top image, are the ends of chromosomes and do not have important genetic information, but serves as caps that protect the genes on the chromosomes. During DNA replication, the ends can become shorter over time, eventually affecting genetic material. An enzyme known as telomerase has been found to lengthen telomeres by adding nucleotides to the ends of those telomeres. This is important in medical research as it could help decrease or even reverse the aging process, as well as decrease cognitive decline in dementia patients and improve functioning of certain cells in the body. If you are wondering about the chances of how you can improve your telomere health, it is possible to do so. Watch the linked video for more information. In prokaryotes, DNA synthesis proceeds from a single point of origin. From that point, in both directions, two replication forks form. Notice the black arrows. Then it travels around the circle and eventually meets to form two separate chromosomes. This process is much simpler than in eukaryotic cells and can therefore happen much faster. During DNA replication, errors can occur and are usually repaired. One type of repair mechanism is called a mismatch pair. DNA polymerase will fill in a mismatch with the correct nucleotide. When ultraviolet rays cause genetic damage, DNA polymerase will cut out the damaged sections of the DNA strand and replace it with the correct sequences. When these repairs are not fixed by the replication process, the individual can actually develop skin cancer. When an error is not corrected, it leads to a mutation. Some mutations are beneficial and can even improve the survivability of a species. However, some mutations can be detrimental, such as when cancer results from a mutation. Proteins are necessary throughout the body to perform all functions of life. For the body to create proteins, DNA, which holds instructions for life, must be rewritten into the language of a protein. To accomplish this, DNA is transcribed into RNA, which is then translated into amino acids, which can then form a protein. This is known as the central dogma of biology, where DNA encodes RNA, which encodes proteins. As a reminder, DNA is a double-stranded molecule, has a pentose sugar known as deoxyribose, which is lacking one oxygen at the 2' carbon, and it has a nitrogenous base known as thymine. While RNA is single-stranded, has a pentose sugar called ribose, and does not use thymine, but rather uracil as one of its nitrogenous bases. The process of transcription rewrites the information in DNA into messenger RNA, also known as mRNA. This process has three phases: initiation, elongation, and termination. During initiation, an enzyme known as RNA polymerase attaches to an area known as the promoter. This area is upstream from the region to be transcribed and is used to recognize the gene which will be transcribed. Once the gene is recognized, the DNA will be unwound and transcription will begin. During the elongation process of mRNA synthesis, there are two strands of DNA. One is used as the template to make mRNA and therefore is known as the template strand. The other strand is not used in mRNA synthesis and is known as the non-template strand. The newly formed mRNA and the non-template strand will be nearly identical with the exception of the nucleotide exchange of uracil with mRNA. The gene being transcribed will have a termination sequence which tells messenger RNA to stop being synthesized. The newly formed single-stranded mRNA will then separate from the template DNA strand. Before the messenger RNA leaves the nucleus on its way to the ribosome in the cytoplasm, the mRNA must be processed. Genes have many codes on them, but not all are protein coding sequences, and therefore some of the areas must be cut out or spliced for the mRNA to be properly read by the ribosome in the next part of protein synthesis. Areas of mRNA which are cut out are known as introns, while the portions that are retained are called exons. Think of introns have to stay in the nucleus while exons get to exit the nucleus with the mRNA when it heads to the cytoplasm. Once these areas are spliced, the mRNA receives a 5' cap on the 5' end, as well as a poly-A tail on the opposite end. This tail is many adenines, hence the poly-A, and serves as a protection layer in addition to a signaling mechanism that it can be exported to the cytoplasm. Once messenger RNA enters the cytoplasm, it will be further processed by a ribosome to translate the RNA message into an amino acid sequence, which then becomes a protein. Similar to synthesis of messenger RNA, the process of translation, which then synthesizes proteins, has 3 phases: initiation, elongation, and termination. Ribosomes are found in the cytoplasm of a cell and have 2 subunits. The large subunit sits on top of the smaller subunit. It is in between these 2 units where mRNA passes through and is translated into amino acids. An important part of the translation process is transfer RNA or tRNA. This molecule transfers amino acids and has a 3-letter sequence called an anticodon, which will pair up with a complementary codon from mRNA. Messenger RNA is fed through the ribosomal units. It is read in a 3-base pair sequence known as the codon. Notice the 3 letters in the pink boxes. These are codons and will be paired with a complementary 3-letter sequence on tRNA known as the anticodon. Notice the letters in the blue boxes. Look at the 2 sets of letters and you will notice they are complements of each other. The mRNA strand will recruit the appropriate tRNA, which carries a specific amino acid. Together, these amino acids string together, forming a peptide chain, which eventually becomes a protein. For translation to occur, specific sequences are required. Eukaryotic organisms use the 3-letter sequence of AUG as a start codon. This codon also codes for the amino acid methionine. There are several stop codons that may be used. Once a stop codon is read, a release factor signals the ribosome to end translation and release the polypeptide chain. The start codon represents the initiation phase of translation, while the stop codon is required for the termination phase. While the ribosome works to pair the mRNA and tRNA to form the long string of amino acids, this central part is representing the elongation phase of translation. Each amino acid needed is determined by mRNA's codon sequence. The complementary tRNA anticodon will carry the appropriate amino acid for that sequence. This process was originally dictated by DNA, which was transcribed to mRNA and now translated into amino acids, which will become proteins that perform the functions necessary for life. Notice there is only one green codon, which is AUG, the start codon, and it codes for MET or methionine. Notice the 3 stop codons as indicated in red. They do not code for an amino acid but are necessary to release the string of amino acids from the ribosome. Many amino acids can be coded by different but similar codons. For example, LEU or leucine, outlined in purple, can be coded by any codon which starts with both letters C and U. Watch the linked video for an animation of the transcription and translation processes in a cell. If all DNA is copied from a single cell or the fusion of 2 gametes, how do different tissues perform different functions? Why do we not have eyeballs in our stomachs or taste buds in our eyes? The process of gene expression and regulation control what genes are expressed. With gene expression, 2 identical cells can turn on or turn off genes in order to perform necessary functions. In the top cell, a neuron-specific gene allows the production of neuron tissue, while in the bottom cell, epithelial-specific genes create epithelial or skin cells and tissues. Gene regulation is the process of genes being turned on or off as needed for life functions. An example of gene regulation is the lac operon in bacterial cells, which can use the milk sugar lactose as fuel. When lactose is present, bacterial cells must produce a protein that can break it down. In order to make the protein, the cell must transcribe the genes from DNA to messenger RNA. Notice in the top diagram, when lactose is absent, the cell does not need to produce proteins that will break down the lactose sugars, and therefore transcription is blocked by a repressor. However, notice in the bottom diagram, if lactose is present, the lactose itself will bind to the repressor, which then releases it from the area, allowing transcription to occur. Once all the lactose in the cell has been broken down, the repressor will once again bind to the DNA strand, blocking the transcription process. So why do you think these simple cells need to regulate the process of breaking down lactose? Even the simplest of cells need to conserve energy, and this is an example of one way a bacterial cell can conserve energy. If lactose is not present, it does not need to waste energy producing proteins for breaking down that lactose. So we are at the end of Chapter 9. A couple of reminders for this coming up week. This week's virtual lab, which is over transcription and translation, is not difficult, but you do need to be prepared to spend a little bit of time on it. It has quite a few questions and several steps to go through. Also, exam 2 is coming up this week. It is available, or it will be available Friday and Saturday, just like before. It covers chapters 6, 7, 8, and 9. Same rules will apply. You'll have 90 minutes. It'll be worth 50 points. If you've not yet done it, your karyotyping extra credit is due no later than Saturday at 11:59 p.m. and 0 exceptions. No credit is awarded if that extra credit is turned in late. Now, only 2 chapters left and only 1 exam left, so hang in there, and if you have any more questions, keep on emailing me.